Hi students, how are you? Let's start with prokaryotes. It's a very interesting topic, and I'm sure that I've already taught you a little bit, maybe from a different direction. You can see the reference of the slides, so it is easy for you to refer, right? Let's go ahead with the lecture. Whenever we say prokaryotes, most of the time we are talking about bacteria, right? Bacteria are everywhere, everywhere. They are on your nose, they're on your hands, they're on something that you touched recently, right? And this is very true. At the deepest depths of the ocean and at the highest point in the atmosphere. They're everywhere, right? And based on this sheer number, and believe me, this number is in billions, much more than us, they are actually the most successful group of life on the planet, right? And one of the reason for this is that they asexually reproduce, right? We will soon know what is asexual reproduction, but it is the fastest way of transferring your genetic material. Now here on the right side, let's play a small game. Do you know which movie? I've written it there. Uh, do you remember this alien machine that was killing all the humans to get their blood from War of the Worlds, Tom Cruise movie? And do you remember the ending of the movie? I mean, it is a very, very good, information for us that bacteria killed these aliens because they don't have bacteria in their world, and the millions and billions of bacteria that we humans are used to, the same bacteria killed the aliens in the movie. So this is one good way of remembering. You must watch the movie. It's really weird and crazy. Okay, on the left side of the screen, you see a bacterial colony, right? This is a colony. This is not a single bacteria. It is a colony. And this is a colorized image to show you how the colony progresses, right? So this is just to give you an example of how a bacterial colony would look when colored with different dyes. Now, the classification for prokaryotes usually always is shown as a 3-domain system. So if you look on the top image, look at the top image, you see a common ancestor written on the bottom, right? And then you see 3 lines. First you have 2 lines, and then the second line bifurcates, right, and gives us 3-domain classification. The first domain, which is also written or known as bacteria, also called eubacteria. The second domain is called archaea, also called archaebacteria, and the third domain is called eukarya or eukaryotes, that are organisms that have a nucleus. So you see that domain bacteria or eubacteria has completely separated, right, from the other 2 branches, which are much more closer in evolution, right? These lines show you evolution, and the further you are on these lines, that means the most different you are. So domain archaea and domain eukarya are still much more similar on genomic level than domain bacteria, right? And in the bottom image, you see now much more information about the 3 domains. In domain bacteria, now you see spirochetes, chlamydias, gram-positive bacteria, cyanobacteria, and proteobacteria. I worked on cyanobacteria, by the way, and they produce toxins which infect your water, right? And people can die from this too, not just COVID-19. So cyanobacteria are also very cool. They are everywhere. Then domain Archaea has extreme thermophiles, methanogens, and extreme halophiles. All these are found in extreme environment. Extreme for us in a way, right? Not extreme for them because they are thriving there. And then domain Eukarya or eukaryotes, where we belong, right? So if we look at Archaea, they are also called as extremophiles, which means that they live in extreme environments. What are these extreme environments? Such an environment is shown in the picture. This is a hot spring, very hot water, right? Salt ponds, Arctic ice, right? Deep oil wells, they even survive in deep oil wells. Acidic ponds, acidic environments, right? So, one thing that we must remember here is even if these temperatures or these acidity, all these environments are extreme to us, they are not extreme to the extremophiles. So life is a very, very relative thing. Just as nowadays when we have lockdown, the earth is flourishing. I mean, I have seen so many beautiful birds come and sit on my balcony because there's nobody they are afraid of. So life is a very, very relative thing, right? For some condition is good, and for the others, it can be not so good. It can be extreme, right? So this picture was of a thermophilic bacteria which loves heat, right? And you can read about this later, but the important thing is that the thermal gradient in this flowing water is actually between 60 to 100 degrees, where all these archaebacteria survive and flourish. So prokaryotes are everywhere, right? As you see in the picture, the boy is a little bit afraid, but actually he's carrying all these bacteria all the time. There are many places that he doesn't really think about other than his hands. His nose, his skin, his throat, right? His gut microbiome is a huge thing, right? So bacteria are everywhere. Prokaryotes are everywhere. They're part of our system. In terms of metabolic impact and numbers, they definitely dominate the biosphere. So we are not the dominating species on Earth, it is prokaryotes. And there is, they outnumber all eukaryotes combined. They live in a myriad of environments, just as I told you before. And even a teaspoon, right? What is a teaspoon? That's nothing. Even a teaspoon of common dirt can harbor 100 million or more bacteria. Just imagine that. This high number is also due to the high mutation rate, because they are able to mutate very fast and they are able to reproduce asexually. So if we go into scientific facts, then 1 teaspoon of soil could reside up to 1 million different species. We're not even talking about the same species, we are talking about different bacteria, prokaryote species. So how cool is that, right? And I will not fit into 1 teaspoon. So we are much more less than prokaryotes. This is all due to the fast growth and high rates of evolution, right? Which means a prokaryote can actually divide in as little as 20 minutes. So 1 becomes 2. And this is done by binary fission, which is shown on the right side. You have a bacteria molecule, no nucleus, you don't need a nucleus, right? Prokaryotes. So the bacterial chromosome is right there. It replicates and gives 2 cells. That's binary fission. Why it is so easy to do binary fission? One is prokaryotes have a small genome. This bacterial genome is much smaller than our genome. So this whole process of replication does not take so long. The other is they have simple morphologies. True. You don't see any mitochondria, you don't see any ER, you don't see very difficult organelles, right? They don't have a nucleus. So they don't need very high-tech proteins to do the job. And therefore they use binary fission, just simply pinching off of the mother into 2 new daughter cells. Okay students, so this was a very fast lecture I guess in total, but we will continue with this, and I just wanted to remind you that before I go forward I have to also do it very nicely so you don't take any topic out of my presentation for your assignments, right? The genetic organization of prokaryotes and eukaryotes I will not be doing because I have given it to a group which is listed on the bottom for assignment. Now how would you do this? How would you do this fun thing? Exactly like how I do. So you make a video and you include your voice in the PowerPoint. How would you do this? First, all the roll numbers should make their individual PowerPoint slide and then add your voice to your PowerPoint slide. So we will do this through this YouTube link. I will play it after this lecture. Once you have added your voice to this PowerPoint slide, add all the PowerPoint slides with the voice insertions into one group, right? And using the Save As button, make a video, right? Just as this video, and then you present this presentation to the class after midterm. Alright, so it's pretty easy and I will play you this video now. Hope you enjoyed this very short session, but I guess next time it will be even more interesting. So bye.